wasn't completely installed, but there was stuff. Okay. This is so close. Very close. All right. Since I have work for Friday. Yeah. Okay. Yeah. If you guys just want to, if you guys can't do it Friday at 10, because I'm sure you guys know, like I'm busy too. So like making time to do things. I usually most of us professors we don't have class on Friday. You guys know that. So I need to do my cultures on Friday to get ready for you guys. It takes days to get this stuff ready. Which is why we don't have lab the first week. It just takes me forever just to get this stuff growing and set up. But yeah, so my point is like Fridays are usually like for that stuff for me. So like 10 a.m. is like I feel like not too early, but I can at least get everything done in one big old go in the afternoon. So that's why I have it set that way. It's just kind of what works for me. I have so many classes that I teach and so many labs. That's like it, man. But yeah, so if you guys can't make it, to the Zoom. I prefer that you try to if you can, but if you cannot, there will be a recording. I will be posting it online. If literally nobody shows up to the thing, then I will still record something. But there's the benefit of you guys having questions about what I'm talking about, you know, because if I'm explaining it and it's just complicated still, like that's not any benefit to anybody. So, right. Yeah. If you can do, if you can't, try to watch it afterwards. And you guys know about how, here we go. On YouTube, like after you've uploaded a video onto YouTube, it will close caption automatically, like it's like an automatic thing. And you can like search within the closed captions for something. So if you're more interested in like me talking about a specific thing, you can search in the lectures and stuff that way. If you didn't know. And of course, watch them double time or something if you can understand. I already talk fast, so it's gonna be hard, but yeah. You don't need to create an account at all. And I'll actually walk you through that here in a second. Yeah, yeah, I'm about to put it up. It makes, I feel like it makes it fun. When we do the reviews for the actual exams, we get this code so that we can get that part up first. So the code for this one, if you guys are trying to join us online, sorry, I'll put it up for you guys in a second. The code for this, if you guys are trying to join online to play, is 517-7151 if you want to join. 517-7151. And I'm going to move this window. That's why I'm telling you right now. Right here. So you guys, all you have to do, if you haven't done Kahoot before, you can go to kahoot.it if you're going to the website, and then just enter in this PIN number, and then that will let you join this game that we're actually playing. Or you can use the QR code, and it'll just take you there. If you're using the app, you just go type the code in, and then you can join that way. You do not have to use your name or your real initials or anything, especially in here. Like, no, it's not for a grade or anything. And if you don't think you're going to do good on some of this and you don't want people to know that you're not doing good, then, put your name as, even carrot, dinosaur, bacteria or something. So I'll let you guys kind of work on that. You guys have any questions for me so far, trying to get oriented with the course? We will have lab on Tuesday. So be sure on Tuesday, closed toe shoes and lab coat. We'll go over, Tuesdays is just like orientation, basically. There's not a pre-lab for Tuesdays. So we'll kind of, everybody should just show up on Tuesday to make sure you have your lab coat and your shoes, of course. But, and we'll go over kind of how the lab is set up, and all of that stuff. Yes. No, absolutely not. Well, like even like the snap ones for the nurses, that's fine too. Yeah. I don't know. I don't think there is here. Has like, yeah, you can buy them, right? But I don't think you can rent them. I've never heard of renting a laptop. You can. I can tell you this. If you if you look on like Amazon and stuff, there's cheap ones. They're pretty cheap. Yeah, I would get it. Amazon or Walmart. But yeah, no, as long as it is a lab coat, that would be okay for general labs. It'll be fine in our class. It says like in the safety that it's supposed to be like flame retardant or something like that. We just FYI, we're going to be using Bunsen burners every single day. So yeah, when it says you need to have something that's not flammable, as long as it's mostly polyester. If it's like half polyester, you're probably fine, or more. If it has any stretch, you'll be fine. So I wouldn't worry about that. But yeah, if you're walking in there like just straight up like linen, I don't know about that because sometimes people just don't think and they'll reach across that flame or whatever. And that's why it says to like have, if you have long hair, and my hair's getting so long now. But if you have long hair, and I would say if it's like, I don't know, about this length, this is about the line that I would draw. And if you're going to be leaning over and your hair could potentially be hanging farther than your face is, then I would be worried that it could get in the flames. That's on accident. It has happened. That's why there's a rule for this. It really literally has, not to me, but in Dr. B's class. So just don't do it. I'm trying to think what else before we get started. So yeah, everybody sort of, if you guys are online, and I know I already said it, but just in case you're trying to play catch up and you're looking at this weird screen, because my screen, this is a different screen, in case you guys didn't know, and that's what they see. 517-7151. I'll probably actually share the other screen. That's where the slides are going to be anyways. I don't know what this is. I don't know what that means. Okay, that surprised me. Let's see. If you haven't contacted your groups yet, which, you know, most people haven't, because I'm not gonna lie, if you're texting each other on phones or whatever, I wouldn't know. But when you guys start chatting in your groups, it creates the group like officially on Canvas and it lets me know. So I know that most people haven't started working on the thing. That's okay, right? You have about I have an alert going out on Monday just to remind everybody that you will have, as of Monday or Tuesday for you guys, about 2 weeks to work on that project. So it's kind of a little sooner than people realize, but yeah, it's like every 2 weeks we'll have one of those projects, every 2 to 3 weeks. But when you're working on those, you should not have any need to like go to outside resources to get more information. So the information that's actually going to go on your projects, you should be able to get it from like, you know, lecture or textbook or study sheet. You don't need to go beyond that as far as content goes. It's not going to impress me. It's meant for you to help review what you need to know for the exam. So there's no point in going past that. If you want to, you know, of course, go outside of resources that we have to get like pictures or something like that, something to help it look funner, then go ahead. But you don't need to cover more information. Okay. And yeah, you guys will be presenting it in front of everybody that day before the class, before the exam. You know, take 5 to 10 minutes to just come up, oh, hey, this is the poster we made, or talk about the handout that you designed or whatever. And it's talking about germ theory and how, you know, Pasteur designed his experiment, and just kind of overview of that stuff, whatever it is that you guys decide to do. So we'll have all 6. You guys have 4 groups in your class. Yeah, all 4 groups. Yeah. however you want. Yeah, I don't care how you do it, as long as like on that day, you guys are kind of working together to present it. Yeah. I know this is random, so maybe you don't want to hang with them, and that's okay. All right, so let's get started with this. This is chapter 3. There are 2 questions on this one. One of them, we just aren't covering the content. I just forgot to change it. And then the other one is like just more info than you need to know right now, but you will need to know it later. Just letting you know, if you get those wrong, that's not on you, that's on me. Okay. Chapter 3. And if you want to do chapter 1, it is available on the modules. All right, which of the following is not included in the 5 eyes? All right. So yeah, most of you guys got this one. I don't know how many of you want to, look it up in any notes or anything, or if you just remember. If you do, just remember, hey, you learned a thing because you probably wouldn't have known that before coming into the class. So congratulations. So it's infection. And remember, these are actually steps that go in order. And when you are familiar with the terms, that makes sense. So you had inoculation, putting your bacteria on fresh media. That's inoculating it, starting a culture, right? You have incubation, so it's getting it to grow. We have isolation, separating it out, like we said with our throat swab, a whole bunch of stuff there. You want to separate it out, isolation step. Then we have the inspection step, which is where we're doing tests to figure out, does it grow on this media? What color does it change on that media? And whatever else. And then identification based on those results. And it could be as simple as inspection, it could just be running it on a PCR machine, if you can. Let you guys know, magic about PCR is that it is not anywhere near as magic as you think it is. And we'll learn about that in the DNA stuff. Okay. Differential media. What does that do? What do we use it for? This one gives you extra time so you can refamiliarize yourself with the choices. This one will be about differential media. So the next one's going to be about selective. All right. So most of you guys got this one, at least the majority, right? So differential media, remember how what we said was it tells you the difference. This one actually has differences down there. On the yellow, it says highlights biochemical and metabolic differences between microorganisms. I know that we said in our talks, it shows you the differences based on the color changes. But why do the color changes happen? Biochemical differences. Okay. All right. Let's go to the next one. Selective media. Now, let's see if you can find the definition for this one. Kind of get tripped up. Really good job, guys. So it's tripped up some of my other classes. I guess that they got like, how we said differential, it tells you the difference, and then selective helps you select. And I guess they got caught up on those having to be the words. You guys didn't get tripped up by that. You saw that, oh, hey, it's going to inhibit some and encourage the growth of others. So we are selecting for some and not for others. I'm glad that you guys didn't get tripped up by the words there. Good work. Like, I'm not kidding. Most of the other classes didn't get that. So pat yourself on the back. It's funny how classes can be different like that. All right, what type of media supports fastidious bacteria? I mean, obviously, to answer this one, you would need to know what fastidious means. Then you would also need to know what those words are. Alrighty. So fastidious means that you require some nutrient to be added into your media, your diet, whatever it is that you cannot make for yourself, but you have to have it to survive, right? Like our vitamins, we said. So that's fastidious. The thing that it needs in order to live is called a growth factor. And when we have that growth factor in the media to grow the fastidious thing, that media is called enriched. Good work. There it is. Okay. All right, where are we at? Streak plate technique. What do we do that for? Good job. So it's going to be for isolating. One of the things, and nobody did choose this one, but one of the things some people might read is like correctly identifying the microorganism. Might we want to do a streak plate in order to identify an organism? Yeah, potentially. But is that what the streak plate is doing literally? No. So I'm glad that you guys didn't pick that one. So at least there's that. The streak plates we're going to be isolating. It just spreads them out, mechanically dilutes them. Which microscope has a higher resolution? This is one that we didn't do. This is one of them. I forgot. This is one we didn't do. Just make a guess. Resolution means you can zoom into farthest, basically. All right, I must have guessed this one right. You're not expected to know this. This is one that I cut out of the class and I just forget to take it out of here. But electron microscopes, they literally spray electrons at things so you can see actual atoms with that. So it's really cool. But that's what they can be used for. So, and nothing is too small. All right. The diagram that's coming up shows a light microscope, like the ones we're going to use in lab. What is the part that that line is coming off of? We did talk about those. Good job. Do I need to say anything? I don't think so. Good job, guys. That's the objective one. All right, true or false? Stains improve contrast. So yeah, that's true. Good job. That's kind of the whole point of a simple stain, right? Lets you see the cell. All right, gram staining. We didn't go over the details of it really officially. We will be going over the details of it in the next chapter. So you will have to know these. Lab 3 actually goes over this. So make your best guess. A mordant, I'll just let you guys know this since we haven't talked about it. It's like what? It's like mortar, like brick and mortar. The mortar holds the bricks together. If that helps at all. That's one way you can think about it. All right. That's how I remember it anyways. So the mortar that's going to be holding everything together when you're doing the gram stain is the iodine, Gram's iodine. The way it works is, I'm going to go over it in detail later, but briefly, crystal violet, and it gets stuck in the scaffolding, basically, of the bacterial cell walls, okay? And it gets stuck in there. If you have a really thick cell wall, it really gets stuck. If you have only like one layer, it's not really going to get stuck. Turns out. So then, you add that iodine. It doesn't matter how much you add if you've got that thin layer, but it'll hold it in place if you've got the thick layer. Then you're going to rinse it with alcohol. That removes any of the excess crystal violet that didn't stick in place. It didn't get held there. And then you're going to counter stain with safranin so that you have contrast. You can actually see. Because otherwise, if you have the thin and you had crystal violet The iodine isn't going to hold it in place. You rinse it out with the alcohol. What color are you? You're clear. You don't, no color, right? So that's why we do the counter stain with saffron. Yes.
What they use in like CT and MRI.
It's made of the same compound, but it's slightly different chemical makeup as far as like the exact, liquid that they'll be putting in. But yeah, it is iodine. So yes.
I hate those.
It makes you feel like you're peeing. If you guys haven't had one, then you don't know, but yeah, we do. Anyway, so yeah, so that's how that'll work, and that's how we'll tell the difference between gram positive and gram negative, which you probably don't know what that means yet. That's okay. Chapter 4 will tell you that. All right, acid fast staining. Another one that's a differential stain. What do we use it for? And we didn't talk about the exact details. And it's another one that we will definitely be going over in chapter 4, as well as exercise 4 in the lab. You'll be doing this stain. All right, so the acid, that's okay. The acid fast stain, it's looking for Mycobacterium species. So Mycobacterium anything, whether it's Mycobacterium tuberculosis, want to guess what that causes? Yeah. And then Mycobacterium leprae is another one. These bacteria are kind of slow growing. Mycobacterium leprae only doubles, like makes from 1 cell to 2 cells. It only doubles like once every 48 days or something ridiculous. And you put it in perspective. When I worked in the food lab that I was doing food testing, looking for E. coli and all this stuff, E. coli samples, we could pop them in the incubator at like 7 a.m. and then I could pull them back out to do PCR on them. They were ready by 7 p.m., if not earlier. So that's fast to have that much of the bacteria doubling enough that you could, do assays for it. So not Mycobacterium leprae, leprosy is so slow growing that you can't use media to grow it. You have to use like animals if you're going to be working with it. Anyways, so Mycobacterium. These guys they're called Mycobacterium because they have a special molecule on their surface called mycolic acid. And that is a wax. It's actually a wax. And it makes them really hardy, and it helps them kind of survive conditions that other things can't. So anyways, that's why they're called Mycobacterium, because they're mycolic acid. Acid fast staining actually has a special staining procedure where we're putting stain with the wax, and it is what we call lipid soluble, so it binds wax really well, but not regular cells, right? So it'll bind the outside of Mycobacterium, and because they have the wax, when you rinse it with acid, it'll stay holding on to that stain. So it's acid fast, so it's going to hold on to the stain even in the presence of going to hold fast to the stain, okay? Even in the presence of acid. So it's acid fast, and that's Mycobacterium. All of the Mycobacterium species are like that. They'll be positive in that test. No other bacteria that you need to worry about will. So if I ever am talking about acid fast positive things, you should immediately be thinking Mycobacterium. All right? If you think this is the last time it's coming up, you're wrong, because it comes up a lot. And I usually remind people about it every time, but remember, you're going to get tested over it. So I repeat it a lot. There's a few of them that are like that. Let's talk about tuberculosis just for a second before we move on. A lot of people think that this is like an old world disease. It's something that we don't really need to be caring about, and it doesn't really affect us, it's not really here. But you are wrong, because as it turns out, you're okay. I thought it was my phone. It's okay. Mine tried to talk to me the other day. Tuberculosis in the homeless population. Okay, they live in close quarters and they have very poor health care, very poor hygiene, and so they share the diseases pretty much all of them. That's why HIV is rampant in the homeless population due to drug use, due to unprotected sex, stuff like that. And of course, HIV is going to lower your immune system, so you're more susceptible to other things. Even easier to get tuberculosis now. When I worked at Southwest Medical Center in the lab, we got a lot of these tests that would be sent up for us that would be asking for an AFB. And it would be sputum. So a lot of homeless guys would come in, and then we would get sputum from these homeless guys and have to run an AFB on them. An AFB stands for acid fast bacilli. So it's literally just doing the stain directly on sputum. And so we would be doing that same right on the field. We didn't have to culture it or anything. And we could see if it was positive for that. Because if it's positive in that stain, it is tuberculosis. There's nothing else it could be at that point, right? That's how kind of almost completely definitive it can be with that test. And so even if you think that maybe you're protected because you aren't homeless, and so you think, good, I'm not going to get tuberculosis though. There was not too long ago, I want to say a couple years ago, when I started working here, it's been 3 years now for that. So maybe a couple years ago that they had 14 people at one of the Edmond high schools test positive for tuberculosis. And they are healthy people. And one of them was faculty and the rest were just students. But yeah, they had tuberculosis. Were they all coughing up blood and dying? Absolutely not. Because most people that have tuberculosis are just carrying it and they don't even know they have it. So the next time that you guys work at a new hospital or healthcare clinic or whatever, and they have you guys get that skin test, they're testing you for tuberculosis. They're testing you because you might not know that you have it and they don't want you giving it to patients. So that's what that's about. We'll learn how that test works in chapter 17. Anyways, it gets worse. Okay. Maybe not for us here in the United States, that's about as bad as it gets, but for the whole world, to put it into perspective, 1/3, I shit you not, 1/3 of the world has tuberculosis. So if you think it's an old world disease and it's gone and we shouldn't care about it, like I said, you're wrong. It is absolutely a serious issue. Now, it's not as common here in the US. We obviously still have it, but we're talking about third world countries mostly. And we're talking about places where they are not maintaining control with antibiotics. I'll ask you guys if you can understand that a lot. There's a pretty high rate of antibiotic resistance with these, especially with tuberculosis. So let me ask you if you would adhere to your antibiotics. First of all, if you went to the doctor and you had like a urinary tract infection or like pneumonia or whatever, and they just pop you some antibiotics and they're like, be sure you finish this. I would hope that you guys going into healthcare actually finish your antibiotics. There's a reason you have to finish them, because if you don't kill off all of the bacteria with that, it's very likely that they will become resistant. I don't mean slightly, like, it'll be fine. Probably not, actually. Probably not fine. That's where you have things coming out like MRSA, things that are antibiotic resistant. A lot of times it's because people aren't adhering to that regimen. So you might think it's hard enough for some people, especially people who, are defiant for children or whatever, who just don't know, to adhere to, 10 days of antibiotic. With tuberculosis, for your first 2 months of treatment of oral antibiotics, your first 2 months, they give you 3 different antibiotics. The one that works the best, you will stay on for the rest of your 7 months after that. How many of you guys would stick to that? Probably hard, even for an American who can set, a notice on your phone and remind you to take it every day for 9 months. So it's 6 to 9 months usually to treat tuberculosis. Now imagine you're homeless or you live in a third world country. Do you think they're taking those medications to completion? So that's why we have such an issue with the resistance in tuberculosis. A serious thing, especially things that I think people should know about that may not be in the United States, but we should be aware of humans, because all humans are equal, right? We should be treated as such. Here's our little podium, and then we're going to go into a lecture. I have these moments where I get on my pedestal. At least it's talking about what we're learning. I could go into other things like Morse code. All right, so cool. We've seen it. We're happy with it. Everybody proud of themselves. You should be actually, you guys actually did pretty good. Let's do this. All right, so today we are learning about, it should be a review about chemistry. You've all taken it at some points. So hopefully this is something that is just a refresher. And I hope that if it's not, that hopefully I've said something that'll help you remember some of this stuff. All right, protons and neutrons, they come together in the nucleus to make the nucleus. Then around that, we have the electrons floating in the cloud around it. Yeah, the orbitals, they say. The electrons, I'll have you guys remember, are negatively charged. And this is important because as I'm going over the molecules that we're going to get into, because probably the most important thing that I can teach you in the chemistry aspect of things going forward would be hydrogen bonds. So if you were like, oh, that was one of them, you're already thinking it, then you have to get it. If you have to get hydrogen bonds for this class, you're going to get so lost if you don't. So electrons have a negative charge. We know it, but don't forget it because we're going to need to talk about it here in a second. There's an atom. There's the periodic table of elements. Okay, so molecules. Anytime you have at least two atoms coming together of any kind, it's a molecule. When we're talking about O2 or N2, it's a molecule, but it's not a compound. Compound, you have to have different kinds of atoms coming together. So CO, carbon monoxide, would be a small one that would fit here. But if it's only one kind of atom, like oxygens, that can only be a molecule, it is not a compound. Compounds do fall under molecules, though. All right, let's talk about the bonds. We're already doing it, okay? Covalent bonds, ionic bonds, and hydrogen bonds. All right, let's move on. Let's get to the picture about covalent bonds. We're going to start there. Covalent bonds are bonds where you're going to share electrons. And it's fine to say, and it's easy enough to write it down and all this, but let's understand what that really means. Because we need to put all these bonds in perspective to each other, not just have their own category here. So in a covalent bond, imagine that you are going outside to play with your sister. I was not a nice sister, okay? I was not. I was mean. I'm the older one. I was a brat. But let's say I'm going out to play with my sister. Her name's Erica. And Erica and I are playing with a Barbie, all right? And we're both playing with it, and we're sharing that Barbie. That's exactly what a covalent bond is. Okay, so we're sharing the Barbie. Sometimes, though, because I'm a brat and I'm mean, I'm going to hog the Barbie. We're still sharing it, Mom, I promise. But maybe I'm keeping it more. Okay, so if that Barbie is an electron, the one that's keeping it more gets a negative polarity. Okay, are we completely separating off? Not at this point. We're still sharing it. It's just one's hogging it. Very important concept because when we have water molecules, which is of course the solvent of life, that's exactly what's happening. I know it's up on the board, but there's a lot that's going to happen in a minute, so it's not good to be over here. Oh, okay. Oxygen, notoriously electronegative, right? And if you need any help remembering about like kind of what electronegative means and how what's going on and why, I always think of whenever it's being called negative, it's like not playing well with others. It's naughty, right? It's negative compared to everybody else. Nobody else likes this because you're being naughty and selfish. And so that's a negative attribute, right? So it's a negative charge. Oxygen does that, like I did with my sister, right? I'm hogging the Barbie. Oxygen's hogging the Barbie, hogging the electron. So it gets this negative kind of charge to it. Call it polarity because we're still maintaining our covalent bonds here. That's where we're sharing. It's sharing with this guy and with this guy, but it's hogging both of them. So it's being naughty. But this is our covalent. This guy's got a plus because it doesn't get to hold its electron as much, right? So it's losing that negative charge too. The Barbie sealer. Okay. So oxygens and nitrogens are the guys that do this. They're very electronegative. That is the number one most important thing we can understand about water molecules and why it's the solvent of life. Next, we have ionic bonds. Imagine, if you would, that my sister and I go out to play Barbie with one another. My mom's like, go out and play Barbie together, Lauren and Erica. And so, or she's my martial art girl. This is usually what will come out of her mouth, right? So we go outside to play. And what if I just take her Barbie and just take it? Like, I'm just going to leave and go play with the Barbie on my own. She'd probably cry. I wouldn't care because I'm super negative. And that Barbie, you can tell that I'm the asshole there because I'm going around with the Barbie and I left the little girl crying. Okay, so that is ionic bonds. Ionic bonds, you're donating your electron or transferring it, or the other one is sealing it. However you want to look at it. but we're not sharing anymore. It's taking it to that extreme, to the point that we are no longer sharing. I know that the most common example that everybody always gives is sodium chloride, right? Where if I was chloride and my sister was sodium, and I take her electron, like her Barbie, and I run off with it. So now I have an actual negative charge as chlorine, and she has a positive charge, she's down an electron. Okay? She lost a negative, so she's positive now. They are actually able to separate because of that. They're no longer tied together by their electrons being shared, like you would see the covalent bond. They can separate. So if you have actual salt crystals, they're hanging out near each other. A positive and a negative are going to be drawn to each other. We know that. So in chemistry, we know opposites attract. At least with charges. Yeah. So cool. So that makes sense. You put sodium chloride into water. Now what? Sodium, my little sister, that positive charge here, that positive charge is going to be attracted to that negative oxygen. Maybe trying to get her Barbie back from that. I don't know. Then this positive up here is going to be attracted to the negative on the chlorine. So now we've actually dissociated them from one another because of these polarities of water. So that's ionization. When we talk about ionization, that's what we're including. Okay? Cool, because you need to remember it. Because these obviously aren't the only things that ionize. Maybe this is one of the deal breakers with chemistry, but you might have H2O. You guys heard about Schrodinger's cat? Anybody know what that is? It's like you have a box and there's a cat inside. Okay, there's a cat in here, let's say. Do you know? I mean, make a noise with it. Hypothetically, if it's just sleeping anyway, do you know if that cat is alive or dead in there? No. You don't know until you open it up, and it is either alive or dead. So that's Schrodinger's cat. But it could be potentially even both simultaneously. How would you know? Because if you open it up and it's just one or the other, how do you know what's going on in that box? It's a philosophical argument more than anything. But man, this is one of those examples I feel like falls into that category. Water. Because you've seen these before. Water does this sometimes. Yeah? But sometimes this oxygen gets so greedy, it just steals this one Barbie and runs off. So it's got that Barbie, and it takes that one completely, and we're just running out together. Right? It's like when another kid came to play with you, and you're supposed to play nice with Terry from across the street. But Terry, I don't know what, whatever. Terry has Barbies, and so you steal his Barbie, and you and Erica run off to go play with his Barbie, and don't include him anymore. And that's fine. That's stuff that actually happened in my real life. We don't need to get into it, okay? Yes, Terry and I are still friends. So, but yes. So if we have any rates on the streets, fine, right? All right. I always put that negative 5 at O so you remember every time that that's. So it can come apart when it gets greedy, but it can be together like this. That's important. Why is that important? We're getting there. Gotta feel this a story, guys. Okay, we did this, we did ionization, we talked about that crap. Hydrogen bonds. Before we get into pH, probably, like I said, the most important thing that I teach you here is about hydrogen bonds. So we saw our little oxygen down here, our water down here. This guy's negative, this is positive, there's the Barbie situation. Okay, here comes another, couple of people that are playing with Barbies also, whatever. Okay, despite that negative charge that this oxygen has from hogging the electrons, that makes it attracted to the positive hydrogens on other water molecules. This concept I know that we've heard about. It shouldn't be too surprising if you've had chemistry. But these charges dictate probably most of everything that goes on in your cells. It's super important. So when we have this attraction from that electronegative Barbie hogger over here, that oxygen, and it gets attracted to this poor little girl that's crying over here, maybe feels that sad for her. They get attracted to each other, that positive, negative, opposites attract. This bond right there, this attraction, that is a hydrogen bond. That attraction that is holding that oxygen up to that hydrogen, because of that positive hydrogen, is our hydrogen bond. So I've already told you it's usually going to be oxygens and nitrogens that get nasty and hot for worms. Yeah. So that is the truth of it, and it's largely oxygens. Okay. So it's largely going to be oxygens that have this negative polarity, and then because of the other association with other oxygens on their own molecules, these hydrogens having that positive polarity, anytime you've got electronegative atoms like that going on, you're going to have some charge to these guys. We'll create this whole web now of water molecules with one another. Pretty easy to imagine. So that's hydrogen bond. They are just attraction. They are not sharing electrons or donating electrons, nothing. They depend on other bonds doing that. But they themselves, the hydrogen bonds, there's no electron exchange. Okay? You can put that here. This is your electron. We're not exchanging it here. It's all attraction. Okay. Let's do this. Okay. So we've got that. We're definitely understanding it for sure. Here's a better picture than what I draw, definitely, but you get it. And I'm never going to ask you about reactants and products. I would hope at this point that you guys know what a reactant is and what a product is. Product is what's made from the reactants doing crap. Solutions, I'm also probably not going to bring it up a lot either, but I have to say it just to remind you guys. A solvent, I don't know why these are hard for me to remember too. You guys ever have a hard time remembering the difference between solute and solvent? Same, okay? And I have a bachelor's in biochemistry. Maybe my master's isn't in that for a reason, okay? But solvents, I try to remember as solvent has a solve with a V, so you're dissolving with the solvent, and then the solute is things dissolving into it, okay? So it's doing the dissolving media, because it's got the V. As much as that can help, but there you go. And you know what concentration is. It's however much stuff is dissolved in there. And we know water is a solvent, and that it works quite well if you have things that have charges or polarities. What I was saying to my other class this morning, think about it as like when you have these polarities, like water does, the oxygen's the negative, the hydrogen's the positive, or you have these ions that are positive and negative, or we're mixing all these polar things and these charged things together. It is dramatic. There's a lot of drama going on in this situation. Positives and negatives, hydrogen bonds, and all of this. I like to paint it that way because there's a lot of activity going on because of the charges. Because what do you get when it's just carbons and hydrogens? A lot of calm. Because carbon is very, very good at sharing. It won't steal. It doesn't steal. It doesn't hog. Nothing. So when you have carbons bound up with hydrogens, those hydrogens aren't positive or negative. They're just hydrogens. So when you have a whole bunch of carbons, the carbons don't have a charge. When the hydrogens don't have a charge. And the water wants to get down with things that have charge. And only things that have charge want to get down with the water. So easy to imagine. And we've got this chill, which starts with a C, like carbon. These chill organic compounds always are going to have carbon and hydrogens. If it's CO2, for example, that's not an organic compound, okay? But if it has carbons and hydrogens in the backbone, that's organic. What we mean by organic chemistry. So we get those guys that don't have any charge at all. We have the charged guys, like just freaking water, even, okay? We mix them together, and what happens? This is where we get this concept of hydrophilic and hydrophobic and amphipathic, right? I will bring these terms up quite a lot as we go through the semester, but I feel like they make sense. So hydrophilic, like Philadelphia, which is where I got my graduate degree, by the way. And where did I live? And you want to guess where I lived in Philadelphia? West Philadelphia. Okay. Hydrophilic, water for hydrophilic, said your brotherly love. Okay, that's going to be water loving. We all know what a phobia is. It's to be afraid. So water theory, hydrophobic. And then we have amphipathic. I like to think of amphi like you think of amphibians. So amphibians can go on land and water. If that helps you pull that concept out of your mind, it does for me. But you can do both. So when you have amphipathic, you've got both. Right? We've got these concepts. We know that our carbons and hydrogens, no polarity, no charge, therefore don't want anything to do with polarity or charge. So when you mix those guys, the carbon hydrogens, the long tails, the fats, the lipids, the oils, the waxes, lots of long carbon hydrogen tails, they don't mix with water, right? So we know that about the fats. That's no big surprise, but that's why they do that. They are hydrophobic because they don't have any charge. They can't get down with the water molecules. But chemically, they literally cannot even if they wanted to. They don't have a charge to do it. So hydrophobic molecules are non-polar molecules. No charge, that's all that means. Hydrophilic molecules, if I were to ask you, hey, what's the most likely one to be hydrophilic? And all your choices have carbons and hydrogens, and then one of your choices has like an OH group at the end. Okay, there's that oxygen. You already know, man. You already know. They're trouble. Okay. So hydrophilic, we've got some sort of charge or polarity. So we dissolve quite well in water because of that. That is, now we are here, our favorite part. I'm not really sure. All right, so we talked about water and how water is going to be H2O, and it's going to break down sometimes to H pluses and OHs. All the H's and O's are still accounted for. They're just apart now because somebody stole the freaking bargain. So that's why this is happening. They can go back and forth. And you know how chemistry is. I'm sure Dr. Tim or Dr. Singh taught you guys, if you had them for chemistry, taught you guys about that they're kind of constantly going back and forth that way. However you want to imagine it. If you have just an H2O, just let's say we have one molecule of water. One H2O molecule, you have an H plus and an OH. They're equal in amount of each other. There's one H plus and there's one OH. So these are equal in number, like quite literally, okay? If you are equal, this is what we would call a neutral state. Equal H and OH means just like a normal water molecule can be formed back. So this equal, it's neutral. Yeah, I'm getting up to where we're going to call 7 neutral. So neutral pH, in case you didn't know, if I'm saying something new to you, equals 7. So we're starting with that concept. You start adding in H pluses or taking away H pluses from the whole mix. No more equal. Just throwing it up now. So if you start adding an increase in H plus, that means we're going to have an acid. Because we're going to have all these H pluses that have nowhere to go. And so they're just kind of hanging out. Doing like messing up the bond and throwing this whole equilibrium off because there's an excess of them. Anytime you're deviating from this equality, there's extra crap floating around, right? You have less hydrogens, and I'm going to tell you right now, I'm never going to ask you what the OHs are doing because pH is referring to the H's, ain't referring to the OHs. I don't care about the OHs, right? Talking about the H's, that's why the H is there in the pH. So you have a decrease in the H plus in that whole milieu. Now I've got less H's and a whole lot more OHs that would be hanging out. I'm not gonna go into detail beyond that, but this causes a base. Or just remember the other word for something that's basic? Alkaline. Okay? If we're talking about alkalinity of something, we're talking about how basic it is. Okay? So this is where we get these concepts of acid and base and neutral. Just the concepts for now. Right? Now, let's look at the pH scale, shall we? Okay. Let's start with the best of all of it. pH. What is it even? What does that mean? We already know that has something to do with the H's, the H pluses, and how many of them are around. But what it literally is negative log of the concentration of. I'm never going to have you do the math with it, but be aware of what it means. Okay? We have the amount of H pluses that are in the solution. We're taking the negative log of that. So what is that? A log is referring about 10. If you're log, you need to say, okay, we're going to do 10 to the power of something. We're talking about log, where like the 1, we're talking about 1, it would be 10 to the 1. But we're not talking about that. We're talking about the negative log. So that negative is what's turning everything on its head here. So if you've got water, pH of 7, that's 10 to the negative 7. Okay? What is that actually? What is this literally? It is the concentration of the hydrogen ions. So if you have 10 to the negative 7, pH of 7, sorry, this is, I say sorry to you guys, boy, cut my leg. That 7 is our pH. That's where the number comes from, okay? So that's cool enough. If I were to write this out for you, 1, 2, 3, 4, 5, 6, 7. That's 10 to the negative 7. So 10 to the negative means you're going backwards from the number, right? With decimal point. What happens if you have an increase in the amount of hydrogen ions? Right? So increase in the amount of hydrogen ions. Well, what is, we already said increase in hydrogen ions is acid. I told you that, I gave that one away. Let's figure out what it really means. So you know it's an acid. pH of a common acid, let's say 2, for like hydrochloric acid in your stomach. Okay? Well, let's say pH of 2. So pH of 2 means we have 10 to the negative 2 as our concentration of hydrogen ions. You can already see that's going to be more. When I write this out, 0.01, if this was money, I mean, first of all, it'd be sad, but you'd rather have the 1 cent than the whatever this crap is. But they do it that way, you have to, but that's just how many decimal places we're moving. That's all it is. So if you have to write it out, you can. But questions that I might ask you on the exam would be, for example, which of the following has more hydrogen, higher hydrogen concentration? Like the let me speak right now. Which of the following numbers has a higher concentration of hydrogen ions, right? And I'll give a list of numbers. If I were to give you right now a number of 8 and a number of 10, which of these has the higher hydrogen concentration? 8. So it makes sense while we're looking at this. Just don't lose it when you're taking the test. Okay? We know pH scale goes anywhere from 0 to 14. It stops at 0 because that's 1. It doesn't go higher than 1. So I'm talking about ratios here. But so that's why it goes to 0. It's going to go up to 14 because you went back 7. You can only go up 7. 7's the neutral, right? So 0, most acid. 7, neutral. 14, most alkaline or base. Okay? All has to do with how many of these hydrogen ions are floating around. We're just talking about pure water. Should be equal. Should be. Right? to you guys about pH and how, man, if you have these extra guys floating around in there, like you would with acids, how that might affect the water molecules and how they interact with one another. Imagine this. You guys know what that is, yeah? This is DNA, right? We call DNA, we call this shape the double helix. Why is it double? Because when you uncurl it from the helix shape, you have two sides. You might have, let me just do this real quick and then we'll get into what this means. You don't need to know all this right now as far as the concepts. It's just an example to show you the importance of hydrogen. Okay? We'll learn this in chapter 6. Yeah, we're not getting away from it. So, I don't know whatever they are. All right, the other side, just so you know, they're always complementary. G and C always bond together, and A and T always bond together. It's the way I remember that is the G and the C are both the round shapes. So they're always together. So that's where I go. So G, there'll be a C on this one, whatever. These are going to match up, complementary, always. That's nice. The way it's like that is for whenever we are copying ourselves, as well as how your body is essentially going to be. But anyways, that's why they are complementary. It's literally like one is the side that you use, and the other one is like the side you copy from. Anyways, so these guys, why is G binding with C and T binding with A? Why is that what's making up these ladder rungs? I've already used this before, right? Remember that? What did I use this for? Hydrogen bonds. Yeah. So this is attractions between the G's and C's or the T's and A's based entirely on this stupid thing being electronegative and hogging the Barbie, and that one losing its Barbie. So one of these molecules has a positive H, and maybe the other one has a negative O, and they're just attracted to each other across the way. And based on their literal chemical structure, they favor one or the other, right? So I'm telling you right now, your entire, of all of your DNA, your double helix of all of your DNA is held together by hydrogen bonds. Pretty impressive. You're not sharing electrons or anything, it's just based on charge and attraction. All right? What's another molecule that's kind of like this? We'll talk about this. This is DNA. I know we're going to come back to this why I'm labeling it. I don't know what it is. Let's talk about another molecule that's kind of similar, important for us, right? I'm not going to make you recognize it. I'm not going to make you draw it. Nothing like that. But does anybody know what this is? It's an amino acid. Yeah. Yeah, probably easier to know it if it wasn't drawn by my from scratch. But there it is. So this is an amino acid. You guys know what they do? What do they make up? Proteins. Okay? So amino acids are going to bind literally this unit to the next one, to the next one, and so on and so forth when we're building proteins. Okay? These, look at that, there's N's and H's, and there's O's and charges. So those guys already, that's what's determining the bonding between the next one. The N's having that like issue, and the O's having their issue, and that's what causes them to bind together. The bond that would happen to the next one is known as a peptide bond. You get enough of these, whatever, and here's another one. Okay. You get enough of these in a long string, and you've got a peptide. Anything beyond 20 of them, and you've got a polypeptide. It's all the same thing, just getting longer. Right? Okay. This, the R word. What is that? It's not a chemical structure. R isn't something. R means it can be any of these number of things. So it could be just an H. It could be this whole mess of this ring of stuff, like phenylalanine. It's got this whole ring of carbon hydrogen that's going on. So all sorts of stuff can be in the place of that R. There's 20 different amino acids that we know of in biology that we use. 20. So that's just like 20. Just here, so get that from that good. 20 different amino acids: alanine, phenylalanine, arginine, glutamate. You guys ever heard of monosodium glutamate? MSG? Amino acid. It's break it to you, that's all it is with one sodium. It is not actually a scary thing, but it can be bad for like your blood pressure and stuff. So, that's too much of it, right? So, anyways, makes these taste good though. Here we go with our amino acids getting linked up to one another via their peptide bonds. They're all bound together, amino acid after amino acid after amino acid. Some of these guys might have a charge, might be polar, some might actually have a charge. Some of them are polar, positive or negative, and some of them are hydrophobic, right? All right, when you have proteins, you've got this whole mess of amino acids lining up. It's going to come up in the slides in a little bit, but when you have this whole line of the amino acids, just the amino acids, that's the primary structure of the protein. It's like having a ribbon if it was just made of amino acids. Just it chilling, primary structure, right? If I'm putting you, who is hydrophobic, next to another hydrophobic, next to another hydrophobic, well, suddenly the body is mostly water. Yeah? Your cells are mostly water. So they don't want to hang out in the open. They want to hide. So what your proteins will do, what your ribbon will do, it will start to fold in on itself to hide the hydrophobics that are not charged, the non-polar groups, away from the water so they don't get in a fight. Right? They'll hide them away. And then once it has the charge, well, negative doesn't want to be with negative, really. It wants to be with positive. Opposites attract. We've got that going on too. Hiding the hydrophobics, having the negatives and the positives be attracted to one another. All of that is going on in your long, thousands of amino acid long ribbon. So we got curling up to hide. We've got some of them that are just wanting to be exposed. Some of that it's a long chain of cool. It's fine. You get these structures that'll form. Some of them will be an alpha helix. Some might be a beta pleated. These are happening just because of these guys next to each other not wanting to be next to each other or wanting to be. Okay? So those immediately next to it. Secondary. Then you can imagine, well, you've got these curled up areas of your ribbon. You have these guys that are all just chilling out. These ones that are looking for a mate somewhere, like positive charge. There are a whole slew of positive charges. They're all looking for negative. Well, turns out maybe there's another slew of negative charges on the other side of that whole ribbon. So now, after we've made these little things from the things next to us, now we can start bending those things around, and they can get involved across the way. We've got this whole ribbon, it's all like tying itself up, basically, because of hydrogen bonds are not, right? That's all that's happening here. Hydrogen bonds are not. So we're creating that ribbon. So when we have that whole structure, polypeptide structure is done. The whole ribbon is done, folding up for itself, just for itself. Turns out when you're a protein doing stuff in the body, you aren't just one ribbon folded up. You're usually ribbons coming together that have been folded up, and they work together to make it work. But subunits, I can say, subunits of them, enzymes and stuff. They come together and they work together, like pieces of building a car. You know, you don't have a car to have a wheel. You have to have everything come together to work. So this is like the wheel, and this together is the car. That's the quaternary structure. There's 4 levels of protein. Just the amino acid sequence. The secondary is going to be the helix and the beta sheet, and the things just immediately next to them. After that transformer, now you get that bigger shape of the whole ribbon. That's the best way I can describe it, honestly. But the whole bigger shape of that one ribbon that polypeptide, creating its shape. And then now it's done. You just had it, feed a wheel, and now we're gonna get the rest of the part of it, the axle, the doors, and whatever else you want. Okay? That's the quaternary. All right, so why am I bringing this up? Because I already told you about how important the hydrogen bonds here are for making the DNA. All of your genes, every single one of your genes, either codes for a protein or has genes that control the expression of those proteins. Not anything else you code for. You're like, oh, I have blue eyes. That's proteins that did that for you. I have the brown hair. That's proteins that did that for you. You're as tall as you are because proteins did that for you. So now, it's starting to look a lot more like those 20,000 genes that you're toting around. That proteins are a lot more important than you thought they were. So proteins are doing everything for living beings. I mean everything. And so if your R groups are, water level, they're charged. They're happy. They want to go party with water. And they're ready to get down. Now they're folding up like they should, and they're making the car that they're supposed to make. And the Tesla's running fine. You start introducing too many of these hydrogen ions that have that positive charge. Now, I would say, excuse my language, but I really don't know you. You're fucking up all this. Yeah? Your ribbon can't fold the way that it's supposed to because those H's are interfering with who's supposed to bond with who. It's like you thought that the guy that you liked at school was into you, but then a new girl came in and she's way hotter and he's not interested in you. That messes up the plans. So that's what's happening when we have too many or too few of those hydrogens. We need to keep them where they're supposed to be in our biology for what we're supposed to be. Each animal can be different with that. Ours is about 7.24. We are very strict on that. You get too high or too low on that, and you've got serious problems because of that. That's why. You're breaking up your proteins. They can't do what they're supposed to do. If you have a seizure because your pH is off, it's because your proteins aren't working right in your brain because of that. And if you look at your DNA, I didn't go into it too much, but you have too much H's there. What happens to your DNA structure? It'll break down too. Those weak bonds, they're weak. You get too many H's floating around where they shouldn't be. It's not going to stay together like it's supposed to and do what it's supposed to. So that is the importance of pH and hydrogen bonds. We did this. So now we get to click through like most of this. You guys are thankful for that. You guys remember this, carbons and functional groups. I'm not going to ask you about it. Okay, our first questions. Everybody stand up for me, please. Let's wake ourselves up and get our brains moving. Blood pumping, getting excited. Who do I have? I just want to make sure that I can count for you guys. Yeah. All right, so first question. I am going to call on David. Hey, look at that.
Oh.
You didn't know you were getting into with this. So the rule is, since you're new, the rule is it's not about necessarily being right or wrong. Just try to get something along the lines of what we're talking about and you'll be fine. These points go to your group. You are Bravo group. So if you get it right, Bravo group gets a point. If you are like completely don't know, then I can always open it up to the group and they can still get a point. So, yeah. It's not really meant, this is not a test or something. It's just meant for, yeah, whatever. What's a covalent bond?
Mostly equally or somewhat.
Exactly. So anytime we're sharing the electrons in our bond, good job. You did it. So, and also the rule is, once I've called on you once, I'm not going to call on you again. So I guess technically if I do call on you, you should sit down, but it's meant to, if you want. I find the standing is meant to help wake you up because we're just sitting on our asses the whole time. Okay, this is for Charlie. This is Christy. What's an ionic bond? Christy here? No. My people aren't here. Okay, Angelica, ionic bond. Exactly. Yep, it's that simple. Delta. Point for delta. All right, next one, hydrogen bonds. I'm sure you saw that coming. We have Taylor. You're fine. Oh, you're good. I feel like even knowing that it is a weak bond is knowing about the hydrogen bond, right? That's an important aspect of it. Yeah, based on attraction, right? So that's the important thing with that too. And obviously it has hydrogen involved somehow because hydrogen bond is literally, it has hydrogen involved. That's why it's named that. So you always should know hydrogen, when it is involved in a hydrogen bond, is always the positive one. It's never going to be negatively charged, okay? All right, next one, types of molecules that tend to be hydrophobic. Jenna. That's fine, that works for me. Yeah, do you have any, I just got your point, but do you have any examples? Like, think of one for me. Something that would be water theory, hydrophobic. Just an example from life or something. It doesn't need to be chemical equation or something. You're okay. So an example, I'll let you off the hook here. Don't overthink it, right? Any of the fats will apply, any of the oils, the waxes, any of that stuff, right? You're okay. I know it's a lot when you get called up on. All right. So you were Samantha, right? Or was there all Samantha? You were Samantha? Is Samantha here? Okay, you're Jenna. Okay. Is Samantha here? People have dropped by not taking their things out yet. Okay, Brittany is on there. I don't want to ask her. She's virtual. Kayla. PH. You got it. You can also look at the board. I know that is up there. So yes, you're on the right track. Hydrogen concentration, right? Those hydrogen, free hydrogen ions. So that's what it's talking about. All right, so that's that. I apparently pulled pretty much every single Bravo that I have in here. So it'll even out eventually. All right, so you guys can sit down. We've already talked about most of what we need to say about proteins and nucleic acids. So we'll probably get to skip over those. Oh, we're getting so close. I just want to get through these last ones. All right. Monomer, what we're talking about is the things that's making up the big thing, right? So we're talking about macromolecules with proteins. Their monomers are amino acids. Okay. Carbohydrates. Anybody know the monomer for a carbohydrate off the top of their head? Sugars? Yeah, sugars. We can call it a saccharide. Man, I like never call it that, but okay. Science says to call it a saccharide. You guys know what it means though if you hear it. Here's some pictures of them when they're linked together and when they're not. Here's the lipids. We know these are the hydrophobic guys, and they're hydrophobic, just like oil and water. They're hydrophobic because of those carbon-hydrogen stability, no polarity. Yeah, we already talked about what they are. Here's some pictures of them. Isn't that nice? We'll go into some of these later. There's only one that I want to talk about with you guys, okay? It's the phospholipid. So remember how we talked about hydrophilic and hydrophobic? And I said amphipathic means you're both, but we just kind of walked off into the wild blue yonder after that. All right. Something that is amphipathic, both water loving and water fearing. Say phospholipid. I brought a little head on here because this is our phosphate. It's literally a P with some O's around it. Up here, and then the tail comes off that. It's a really long tail though compared to that. Look at how many freaking oxygens we have here. Not the P that's important here, the phosphorus isn't what's important. What's important is the oxygen, how many freaking oxygens we have that want to steal Barbies. Okay? That's a big deal with a phosphate group. So obviously, this guy is negatively charged here. And then this section down here, the tail, is water fearing, hydrophobic, so carbon and hydrogen. What does that mean for us and why is this important? Why is it here? Okay, so you have your little heads that are charged, and they're charged, and they want to interact with the water, right? That's fine, they're chill because it's all charged, right? But charge makes them water friendly. We're gonna be all right. However, this tail don't want to have anything to do with water, nothing. So, they're going to it like that because they're still gonna be around water down here. So what do they do? More. Imagine this goes on. Now, when I talk to you guys about a phospholipid bilayer, this makes sense now. Because we got phosphos and we got lipids here. The lipids are what here? They want to be together. So they seal themselves up inside of those charged layers on the outside. That's the interaction with water. Water out here and inside. The cells are mostly water too. That's why they will build up like that. And you can imagine that's why your membranes on your cells, if that's your cells have a bilayer like this. That's why your membranes are so good at keeping stuff inside and stuff from out the kidney. Okay? Because who wants to go through that? Who going through weirdo tails and all these charged heads? It turns out that there's only one molecule that can do it successfully and reliably, and that is water. And we're going to talk about water and its importance on how it can cross that membrane when nothing else can. When we talk about osmosis in chapter 9. Okay. But that is the importance of the phospholipid and why your membranes are the way they are. Your cells have the shape that they do because of sterols. That's cholesterol, literally, for us. Okay. Cholesterol is what gives your cells some sort of stability and shape. So they're not just like liquid hot mess. That you can have actual shape to your body thanks to the sterols, cholesterol in your membrane, just kind of throughout it. Other things, steroids and waxes also fall into the lipid category. Anything that would repel water, right? You think about paraffin wax, does that mix with water? No, right? So that works. Okay, we already talked about proteins. What do? They're enzymes. They do literally everything for us and dictate everything that we are. These are those amino acids I was talking about. You don't have to memorize them or anything else about them other than you know that there are amino acids that can have different characteristics. And that's what determines the shape of your protein and therefore the function of your protein. Protein, the actual shape is more important than the chemical exactness. So if you have one charged amino acid, you have a mutation that leads to another amino acid that has a similar charge, you might still be doing the same thing just fine because the shape of the protein is more important, right? We already talked about this, yes. I'm not gonna ask you about that. We did that. We did primary and secondary. There are pictures that are better than my descriptions on here, so I do recommend that you guys go look at the pictures. I just can't do it justice when I draw it, I'm sorry. And we know that proteins, mostly for us, are going to be enzymes. So they're things that are going to help do work in our body. And they're reusable. They don't get used up. And then antibodies, we will talk about what they are in chapter 16 and what they do for us. It's your immune system. Just FYI. I know we're at the end, but I just want to finish this real quick. So denaturing something means you're taking away its normal nature of what it normally does. If you denature DNA or a protein, you can do that with those hydrogen excesses. With acid. Introduce too many hydrogens, you're going to break those hydrogen bonds, or breaking down the structure of what it should be. It can't do the thing it should normally do. That's denaturing. But because these are weak bonds, you can also just do it with heat. So whenever you guys cook an egg, can you get it to go back to its original form? No, you've permanently denatured those proteins. They can't ever go back. Okay? Same. Nucleic acids, you guys know DNA and RNA. This isn't news. We're going to talk about the functions of them in chapter 6, so don't really worry about it too much. But nucleic acids are made up of nucleotides, and a nucleotide has sugar and a phosphate, literally just those little two things. The sugar is a pentose, it's 5 carbon, phosphate, and they just repeat over and over again to make the length. And then in the middle, we have the nitrogenous bases. That's the A, T, C, or G. Nitrogenous, right? And I told you guys about nitrogen. Nitrogen is pretty important. Right. That's really all we need to talk about. I'm never going to ask you about purines and pyrimidines. Another thing that I didn't like in biochem. We'll focus more on the functions of these guys and the structures of them in chapter 6, like I said. This is the last slide. This is ATP, and I know if you guys took chemistry here, you learned about ATP. ATP, adenosine triphosphate, tells you what it is in its name if you know what you're looking at. Adenosine means adenine. That's your ATCG, A. It's the same one. And then we have the adenosine. The sine tells you that there is a sugar there. That's going to be ribose. It's just like the other nucleotides. So ribose. And then our 3 phosphates. So we're talking about how nasty our oxygens are and how they constantly want to steal a Barbie and how they're just like the worst, right? So look at that. Look at those oxygens, you guys. Isn't that crazy? They don't want to be anywhere near each other because they're all negative. Negative people don't want to be near negative people because then they have to face the fact that they're negative. So they don't want to deal with that, but they have to. They've got covalent bonds between them. So whenever you break those bonds, what it is, I don't want to drop my fidget spinner, or else I'm going to break it. What it happens is that those phosphates, they're constantly fighting one another, want to get away, but they're being like chained together, basically, whether they want to or not. You break that bond from one of those or 2 of those phosphates, it releases potential energy. The potential energy is like when you hold this marker here, you might not think there's any energy going on. You guys stand here totally still, but there is. I just released it. So the potential energy that's in those bonds, that's what serves as the energy for your cells. Let me tell you how important this is. Chapter 10 is where we're going to get into it. It is my absolute favorite chapter of all of them. Okay? But briefly, so you understand about ATP and why it is important and how important it really is. I say it's energy for your cells. I mean, it is the energy for your cells. How do we get ATP? It's something called aerobic respiration. Well, again, chapter 10. But it relies on hydrogen ion gradients. So outside of the membrane, wherever, we have tons of these positives building up on the outside, right? Do they want to be near each other? No, because the positive want to be near positive. And anytime you guys have, like, let's say a whole bunch of balls held up in a net, and they all want to come down, right? Same thing, they want to equilibrate, that's the word. They want to be even on both sides. You get all those hydrogens all wanting to come in. Well, they can only go through one door, and that is called ATP synthase. And when they do that, ATP is made, right? But they're coming through the door. How do we keep it with lots of them on the outside and not building up on the inside? So we have to have that gradient in order to power making ATP. It's so important, you guys, that if your gradient is dissipating and we don't keep that going up, so many hydrogens just waiting in line to get into the club or whatever you want to think of. If you don't keep that up, you can't make the ATP. So we have one molecule that we rely on to help take care of this. It takes those hydrogens that are coming into the cell and gets rid of them and turning them into something else. Turns them into water. Do you want to guess what molecule it is that we're introducing? Oxygen. You breathe oxygen only for that. And it is so important for making ATP, the only reason that you breathe in oxygen, to make ATP, that if I suffocate you with a pillow right now, you die because of that. You die within minutes because your body runs out of that so fast. That's how fast your body needs it made. You die from it. And that's something. I feel like nobody understands the gravity of that. So we'll talk about that more whenever we get to chapter 10. That's it. That's all I'm going to spew at you guys for today. Thank you for sitting with me a little bit. Next week we'll be going to lab. Don't forget.
I knew how important ATP is because they talk about a lot of it in biology that I took.
And lucky that you guys take biology. Most students don't have to. So.
Well, I took biology last year before I went to LSU, like changing colleges, and like we talked about more about like all of this, like ATP and stuff like that, than we did in my chemistry class. But my chemistry teacher was two masters bio machine.
Yeah.
I don't know. Fancy.
Yeah.
But 2 masters in it, and he was like 3 times retired. And the way he taught was just.
Yeah. It's like, so many people take the class that way, right? You learn about ATP and like, this is the important energy molecule of cells. And that's kind of where they leave it at. But I want to tie the things together. It makes more sense that way.
She tied them together, but guess I didn't realize it wasn't like it wasn't necessarily like the oxygen was
That, yeah. I mean, everybody knows if I suffocate you, you die.
Yeah,
But most people don't know why. Like, if you go that far,
right.
they're like, wait, because you need it to live. And I'm like, yeah, but why? So that's why. Now you guys know. We'll get into the details of it though. It's a lot of fun. It's my favorite chapter.
And my biology teacher taught a lot like you did, and I really liked it.
It's a lot, but yeah.
It keeps it interesting.
Yep. See you guys. I see. Yep. Thanks, Brittany. Thanks, ladies, for joining us. We'll go ahead and close out of this now. I hope that you got something out of it. I'll see you guys next week.